well, in fact, I would like to be as representative as possible of the pharma industry. Of course, I have a slight bias because I do work for a very small biotech company that has no revenues, right? So since I left the big pharma, I moved into this kind of development. Indeed, I've seen things in a slightly different way as well. And so what I'm trying to do today is to let you know what has been partially my experience, but also what we have learned across a number of different experiences that might be useful for you. All right? So now so the green is to move. Yes. So very quickly, I just wanted to wrap up what is the roadmap for the development of a drug. With a very specific purpose that you're going to see in a minute. So in general, we all start from a medical need and a hypothesis. And this is a very important point, in my opinion. Not having a hypothesis to test is a very dangerous thing to start with. And I want to say that when I think about this, I always refer it to another question, which is very important, and is how much time do we have? Because you can do many things when you have a hypothesis, but you usually have a given time and a given amount of money to get to where you have to. So you have to make choices. And that's very important because it is at the very basis of the experimental hypothesis you want to test, which means that we cannot do everything at the same time, and we have to be very, very careful as to what we pick. A big mistake at the beginning is going to completely destroy the development plan. A big mistake later on is going to destroy it at a much higher cost in any case. After that, you know, a company or whoever wants to move forward a drug has to do all the enabling studies. And I can tell you, if you're not familiar with that, believe me, the non-clinical bit of an IND or a CTA is a huge part. And it must be done very diligently. I go back to the point, experimental hypothesis. The drug has to work and has to be safe. But the first thing, at the very beginning, it has to be safe. So this part, for me, is extremely important. Then you have to design a trial, which looks very easy from the at the first glance. Believe me, designing a clinical trial is becoming more and more complicated, more and more challenging. And I do believe that we have to be responsible developers, have to have the good communication between the caregivers, the company who has the drug there to test, and the regulatory bodies. Because at the end of the day, this is what it's going to be about at the end of the day, looking at the data that have been generated to answer certain hypotheses. But if the methodology is wrong, we're not going to be able to make any decision. A lot of wasted time, a lot of wasted money. But we're going to talk a lot more about clinical study design because I think this is the core of the matter. Then clinical study performance. There are a lot of people that think that once the study is designed and is up and running, this is about it. No. It is complicated to monitor a study. It is complicated to monitor a site where no patients are recruited. There are a lot of people who think, and believe me, I got this comment, we have a huge European grant to develop the anti-interferon gamma. And one of non-European Commission reviewer, an external reviewer, so it could be one of you, challenged us on cost because we were asked to prove that a non-recruiting site generates cost. A non-recruited site generates as much cost as a recruiting site. So the selection of sites is extremely important. And rightly so. The right sites have to do studies, not everybody. And there's a lot more here. And then final data evaluation. The reason I want to talk about final data evaluation is very specific. Because whatever we do for an indication for which either there are drug approved or there are no drug approved, but there are the so-called available therapies, plays a big role in the ability to judge whether the data are good enough to be approved or not. So that's a very important point. So all this goes with monitoring process and regulatory process. So at each step, and depending on which drug you're developing, you may or may not have many interactions with FDA and EMA. And I can tell you, if I may say to the regulatory people here, as we have both breakthrough designation and prime designation, and I think it's so much work. It's so much work. It's a lot more work. It's a lot more work for us, and it's a lot more work for them. But I think it gives a certain reassurance. So it's good stuff to have, actually. Now, what changes when you have to develop and you do all this in a rare and pediatric disease? Well, there are a number of things that do change, and really a number. And I'd like to address that one by one. The one thing that I want to liquidate right away, because it's not the right place, but is to understand that inside the enabling studies that allow you to move your drug into a human being, if you want to develop your drug for a pediatric indication, and say, like, I'm sorry, I have to refer to our case, right? Like in our case, you don't have data in adults. You have to rely on studies, toxicology studies, for example, that are done in juvenile animals. And I guarantee you, this is a nightmare. It's a very difficult thing to do. So this adds, this is yet another burden that you have to think about when you develop a drug only for a pediatric indication. But let's move on. Medical need usually is very high. When we talk about a pediatric and rare disease, there's no doubt the medical need is high. And that's why I use a little bit, the story of the molecule that we would like to become a drug, to guide you through what is the process and what are the challenges, the nuts and bolts that you may find while moving forward. The disease we are trying to address is HLH. I think you heard enough about HLH. What I want to highlight is what has not been yet said, that there is no drug approved specifically for this indication. And it's bad for the patients, but it's bad also for the people trying to develop a drug. Why? Because there is no reference. There are the so-called available therapies that have been not developed specifically for an indication, but there is accumulated knowledge about this. But this is certainly something very important. And in HLH, the vast majority of the so-called available therapy are glucocorticoids and chemotherapy, in particular VP16 etoposide. Now, it's even more complicated because although you treat a patient, if a patient has a primary HLH, it has to go to transplantation to be cured. But you have to bring the child to transplantation, and that's why you have to bring the child to transplantation in the best possible manner. Mortality. What is the mortality in HLH? And that's what defines really the medical need. Well, it's pretty high. And I'm afraid to say that it was high. There's a pointer here, right? It was high when reported by the HLH-94 study. It is, and this is hot off the press, actually, because I took a picture of the... I hope you don't... Well, I told everybody. I told Yang Ying and actually I told Mike. So, in fact, you can tell that I'm not a very good photographer. But the bad news is that from the HLH-94 study to HLH-204 study, a lot of years later, the additional cyclosporine has not improved the situation. The next study hit HLH US and hit HLH Europe is the same, I'm afraid, and this is a study that has affirmed ATG followed by hit the HLH-94, not a great deal of improvement. So is the medical need high? Boy, if it is. Yes, it is. It is very high. And important to say that the drug that you see here, I just mentioned that they are myelosuppressive. They are broadly immunosuppressive, and some of them have also a couple of other unwanted side effects. And this makes things very complicated because if you use a drug that is a myelosuppressive drug for a disease for which pancytopenia is one of the characteristics, then you start wondering, is it the right drug that I'm using, or should I not use something more targeted? And this is the hypothesis. So when we approach, and just to give you an example, when we felt, and a lot of people felt we were crazy, right? As the first drug to develop, we developed a drug for such a rare niche indication. But it's to understand whether or not the same can be obtained in a safer manner. It's not only about efficacy, but it would be great if we could do also that. But I just want to say that safety is also very important. Now, I don't think I need to go into anything like the experimental hypothesis, because I think enough data has been generated to prove that interferon gamma is important. Doesn't matter how, but it is there. It's a bad guy. We don't know how much is bad, but whenever it's there, it's bad. So let's talk about the clinical study design, which is something that is really close to my heart. And in particular, start talking about dosing. And it's very important to talk about dosing, in my opinion, because knowing how to dose a drug is one of the important responsibilities that the pharmaceutical company has got. And I hate to say this, but I really have to say it. When things are done, picking a drug here, a drug there, and then trying for the good to save the life of people off-label, not always we're using the drug in the right manner. And this is very dangerous, I think. And whenever, I'm not saying that it's always possible, but whenever possible, we should look at PK and PD for a drug. So I try to summarize a bit what are the key issues that I encounter when we do so that you also appreciate what are the risks of not doing certain things and why certain people may say, I'm not quite sure I can give the drug to your child because I don't know yet how to use it. So the story in HLH gets even more complicated because HLH is not only a severe disease, but is acute and is life-threatening. Whenever you want to target a certain cytokine like interferon gamma and you propose it against, for example, chemotherapy, it's not that you can go and say, okay, I'll start to give a little bit of the drug and then I'll see how I respond, and then in case I increase the dose. Not possible, because it wouldn't be ethical. So when we wanted to design the trial, what we did was trying to come up with a dose that would have delivered a concentration that would have neutralized what we believe the concentration of interferon gamma in that patient was. The dose, therefore, needs to be adapted because we had so many unknowns, and I'm going to go through the unknowns, and this is what everybody in the pharma industry have to do, hopefully, that we wanted to be sure we could increase the dose, but how to do it with an antibody, which is an IgG1. So it does what an IgG1 does. It stays around for approximately 18 days. So we thought of doing something completely different, and that's why I mentioned before maybe being innovative, allows us to discover more. And what we did is we thought of administering the drug very frequently in little doses to be able to adjust the dose quickly. I'm going to go through this in a second. But also, we suspected that the production of interferon gamma could be very variable in patients, okay? And so we wanted to make sure that individualization of dose was to be possible. So when we designed the trial, we realized that we couldn't do a normal dose-finding study, and we couldn't use the property of the antibody for what it is. It's very convenient. You give it, and then for a couple of weeks or for a month, you wait. That was not the case. The big problem was that also we couldn't use very much data in healthy volunteers because healthy volunteers don't have, if you are healthy, you don't have circulating interferon gamma. So what you learn from a study in healthy volunteer, not very much. And to be honest with you, we went both to the FDA and to the, not to the EMA, but whatever agency we went at that point, I don't even remember. We said, can we go directly into children? It doesn't make sense, to go into healthy volunteers because you neutralize something that is not there. So what do we learn about the PK and PD and safety? But rightly so, they said, no, that's too aggressive. So we went back and we designed a study in healthy volunteer, which we did. We didn't learn very much, as everybody knew, but at least we appreciated a couple of things that then we were able to use. So the first question was how much interferon gamma is produced in a child that we want to treat. Because if we don't know that, how do you know which dose to give in an ethical way? And do the levels of free circulating interferon gamma reflect the amount of interferon gamma produced? So you have a child that presents, you measure interferon gamma, and you say, okay, this is the level, and we looked at the literature, and the literature remember this number, 3,400 picogram per milliliter was kind of the average number for the concentration of circulating interferon gamma. And that was the number we had in mind when we designed originally the study. But because we were not quite sure, we decided to go for a PK-guided study, because we wanted to make sure that in case that number was wrong, we would have corrected it. I'll skip all this because you've already seen it, because I want to go to the conclusion of the story. Well, the story was, yes, in fact, we measure some gamma, but the amount of gamma that we measure at baseline was actually only a fraction of the amount of interferon gamma produced in this patient. And to make a long story short, you see that in the current trial we're performing, the free interferon gamma at baseline is only, on average, at the median value is 50 picogram per milliliter after treatment. And when you're able to drag out of the tissues the interferon gamma that is there, you see the number here, over 17,000. So this tells you how things have to be done very cautiously, because there are a number of surprises when you start treating patients. And if anybody thinks that interferon gamma is produced constantly, it's wrong. Interferon gamma is very, very variable. I have finished my time, so I'll try to go very quickly through remaining. And this is another very important lesson learned as to why we have to be very careful in making assumptions that are only taken from the literature, using assays that, for example, have not been validated completely, because this is the variability of the interferon gamma production that we see in patients, and that's why it's very important to be able to adjust those. And I want to leave you with this very last concept before I move to my conclusive remark on the other part, that is whenever you give a drug, you don't know whether how the drug is going to be cleared. And the drug can be cleared in a linear manner, like what you would expect, or very rapidly, because once you inject the drug, it binds to the target, and then the clearance of the complex is something which is neither like the original drug nor like the target, but it's something different. But you don't know until you try. And because we didn't know that, and I skip all, the ceiling concentration, the lower concentration, but this is exactly what you can see when you give a drug. Either elimination that is linear or a disappearance of the drug very quickly. And this is why we decided to actually dose the drug every 3 days at the beginning, because we wanted to know whether a child was on this trajectory, okay? But I close the story just letting you know that this concentration that was our first target based on literature data and data that, through observation in human being was very wrong, actually, because you saw the amount of interferon gamma that needs to be neutralized is far more than 3,400. And that's why we will ask for a certain indulgence from the people who will have to review our data, because when you see that you don't have 3,000, but you have 17,000 or more, and I can tell you we went up to over 1 million picogram per milliliter of total interferon gamma, you have to understand why adapting was so important. Now, because time goes by, I'm not going to show you real patient data. I think the importance of biomarker can only be stressed in front of this audience, and CXCL9 really in our hands seems to be a very good biomarker to indicate how much active interferon gamma is circulating, and it is our ambition to be able to study this and develop it as companion diagnostic. We can learn a lot. We can diagnose things better, but we also, and why not, it's not a binding statement, but why not consider also for therapeutic drug monitoring? There are a number of things that we can do once we know interfering, when we manage and understand this kind of biomarkers. I'll skip this because Fabrizio is already talking about, and I'm going to conclude on this last couple of slides, just showing you why is it so important when we design a clinical study to think about a couple of other things. First of all, population. For a rare disease, the population is key. The problem is when you have a rare disease in a very in life-threatening, the tendency in a new drug, not a drug that has been already studied in a number of other indications, then the choice of the population is key. Problem is that we're forced to go into the worst of the worst at the beginning. It's a little bit like in oncology. You cannot start a new drug, and give it like this. And this creates a very important problem when you have a rare pediatric disease, because I can tell you that, for example, in our case, an average child has 40 concomitant medications. how do you analyze the data? It's not simple. And this is a problem because if you want them to transfer the data to other population, it's not easy because the child is treated with so many other medications. It also, because you restrict so much the population, you separate yourself against compassionate use requests, obviously. The smaller population, the more people are going to ask for compassionate use. The other important point is the definition of the endpoints. You have no idea in how many cases endpoints for diseases are not defined or not well defined or not validated. And when it comes to designing for the first time a real regulatory term clinical trials, you're facing the problem. And I just want to give you one example. Is this example again of HLH. These are the criteria for the diagnosis of HLH. And what I just want to tell you, that these are the criteria for diagnosis, but there are no criteria for monitoring response to a disease. What is more important than something else? And even hilariously, even more so, for example, soluble CD25. I don't know how many of you can dose soluble CD25, but probably not many. Or this is a very good representation of hospital. But a normal hospital doesn't even know what soluble CD25 is. The reference that is put into the guidelines is totally unvalidated. I don't know for which age range, we don't know. And yet, we are obliged to follow this. But we're following something that is not necessarily right. Another very good example, who does NK cell activity to monitor whether or not the disease is recovered? Not very many. I think this is a problem that's very close to my heart. Ferritin for the diagnosis has to be greater than 500. But we all know that a patient who has been suffering from HLH for a long time has a very high level of ferritin. As below a certain concentration, it's impossible to go. You need months. So if my criteria for definition of response is below 500, I'm going to fail by definition. So that's why it's so important to negotiate these things. And we're learning. We should have negotiated a few things earlier. But this is just a good example of how we cannot take everything that is there if we're not sure it has been validated. And imagine the CNS involvement is not even mentioned in this criteria. So how do we relate to this? It's a patient has CNS involvement from HLH. It's a big problem. I could talk for hours about these things, but I want to close with the last point, which is actually how do we look at our data vis-a-vis the data that is available at the moment of having to take a decision, is this drug going to work or not? I skipped this. It was actually just to show you how hard it is to recruit patients in a rare. I don't know how to go backwards. But anyway, it took us 2.5 years to get 3 patients, almost 3. And I can tell you, you have to beg people to keep paying for what you're doing, paying for the sites or not recruiting, paying for it's a hard job. It's really a hard job. And that's why collaboration with the community that is interested in a certain disease is so important. The context data, and I want to finish on this, is very important because we're all responsible to generate data that are reliable, reproducible. I think there are not enough prospective studies. In the case of HLH, there are a couple of prospective studies, but I can tell you they are either so old or they haven't collected enough data that we can't do anything with that. There's not even a definition of response. And fields are evolving, but this is what we're up to because the results that are there were generated years ago, but we have nothing to compare. And doing randomized trials in these diseases, you know how difficult it is. By far remains the best thing to do, but it's not always possible for a number of reasons. And I want to conclude just saying that in my opinion, this is my personal experience, there's a lot to do in this field. I think I'm very honored. I can tell you that I'm not sure it's the first drug approved for SJIA, but probably yes. I remember the first time I went to the board of the company I used to work for when I said, I think we should develop this drug for systemic juvenile arthritis. And the answer of the chairman was, you must be crazy. It takes a while to convince people that it's worth. And it's worth because we understand so much about diseases. we understand really a lot. And I wish to do a lot more in this field simply because we can understand more about a lot of other things. And this should be the driver, not only for the academia, but also for the pharmaceutical companies. I think, and I close on this, risk sharing is something that is very close to my heart. We have to understand, each of us has a different definition of risk. Risk for a parent that has to treat his or her own child with a certain investigation of drug. Risk for regulators who have to approve, and I have a lot of respect for that. Risk for a pharma company, that puts a lot of money. I can tell you, clinical research costs a lot, and we have to make choices. And people at the hospital, caregivers that have to agree to do certain things. But if there is not a little bit of risk sharing amongst all these stakeholders, I don't think we will move forward very quickly. Because to study well a drug, it has to go in the hands of the physician. So I wish we could do, and there are mechanisms now. There are mechanisms, I must say, to get to the, excuse my expression, to the market. I have no idea how to say that in any other way, but so you understand what I'm talking about. Because it's there we're going to see the good and the bad with a responsible package of data. And that's why I encourage everybody, regardless of the severity of the disease, to study well the drug from the very beginning. I know I've been long, but as I'm the only company representative, thank you very much.